Hello dear students, today in this video we are going to discuss the sample paper of the year 2020-21. In this video we are going to discuss the questions from section A. So section A are very short answer type question which you need to answer in one word or in one sentence, or it can be even multiple choice questions where you will be given different options will be there, A, B, C, and you need to choose one. Or it can be either the type reason and assertion where different options will be given and you have to choose one. So let us discuss the first question. Question number 1, which is why does an endosperm precedes embryo development? So as I tell you that this can be answered in simple one sentence. Answer can be like, because it is a nutritive tissue. Nutritive tissue. Right? So endosperm is a nutritive tissue. So as to nourish the growing embryo, endosperm precedes embryo development. So in this way you can write it simply in one single sentence, that why endosperm development precedes the embryo development. Let us understand what is endosperm in detail. Even you can get the same question in the NCERT book of page number 34, biology class 12. The same question, why endosperm precedes embryo development, or why development of endosperm precedes the embryo development. So let us understand what is endosperm. So endosperm, they divide repeatedly in order to grow their number of cells or tissues, which are called endosperm cell, right, or the primary endosperm nucleus. So they divide repetitively in order to nourish the growing embryo. As we know that the embryo sac, the polar nuclei, which has two cells, are fertilized by the second gamete released through the polyp tube, right. So one of the male gamete fertilizes the egg, which results in syngamy. So if I draw an embryo sac here, the egg is fertilized by the process called syngamy, right. So the egg is fertilized by the process called syngamy, which results in the production of the zygote. Okay, this zygote is diploid, whereas the two polar nuclei, which is also fertilized by the second male gamete, which is known as the triple fusion, right, which is known as the triple fusion. And this fertilization of these two polar nuclei leads to the formation of PEN, or the primary endosperm nucleus, which is a triploid cell, right. So this is the question. The question is why the endosperm development precedes embryo development. Precedes means before. Why before the development of this embryo, endosperm development takes place? So that is our question. And to understand this, we can understand that this zygote, which is going to become the embryo, and this embryo is our future plant, right. This is our future plant. So to get our future plant, or to develop this embryo, nourishment is very important. So that nourishment is done by this primary endosperm nucleus. So here you can write nourishment, right. So the nourishment of the growing embryo is done by the endosperm cell, or the endosperm nucleus, right. So in this way you can write your answer simply in one sentence, or even you can write in two, but these are for short answer type question. Now let us see question number 2, and here is the question: how many meiotic divisions are required to produce 76 seeds in a guava plant? So guava fruit, we know it is a multi-seeded plant, and how many seeds are produced? 76 seeds. Now the question is, how many meiotic divisions are required? So in the question, it means it is asking about the total number of egg plus the total number of pollen grain. That is the total number of male and the female gametes are produced by the number of meiotic divisions. So we have to find out how many meiotic divisions will be required to produce 76 number of seeds, right? So to work it out, first let us see how many male gamete will be produced. Here in this case, we know that one pollen mother cell by undergoing the meiotic division results in the production of 4 haploid pollen grains, right? 4 haploid pollen grains. It means if we divide, it means dividing total number of seeds 76 divided by 4, how much we obtain? We obtain 19, right? 19. It means total number, how many number of male gamete will be required? Total 19. So you can write here total how many number of pollen grains will be obtained. So total pollen grains is equal to 76 divided by 4 is equal to 90 pollen grains. 90 pollen grains. Okay. Now the female egg, how many number of egg? So total, since 76 seeds are produced, it means one embryo sac contains 1 egg. So total it will be 76 eggs will be required, and this 76 eggs will be required, right? So by adding, so total how many meiotic 76 divisions, sorry, not egg, 76 divisions. This one also not 19 pollen grain, I am sorry, 19 divisions, right? Not pollen grain. So 19 divisions to produce pollen grains. Okay, 19 divisions in case of male and 76 division in case of female to produce the female gamete. Okay, so total will be 90 plus 76 is equal to 5, 1 minus 8, 9. 95 meiotic divisions. Right? So total how many? Total 95 meiotic divisions will be required to obtain 76 number of seeds. Right? So in this way we can find out how many meiotic divisions will be required to produce 76 number of seeds in a guava plant or in a guava fruit. Right? So how did we do? First we calculated how many number of divisions will be required to produce the number of pollen grains making 76 seeds. Right? So to do that, 76 seeds divided by 4. Why divided by 4? Because we know that each pollen mother cell undergoes division and produce 4 haploid gametes. Right? So in this way we found out that total 19 divisions. Whereas in case of meiotic divisions in the female gametophile, here we require 76 divisions, result in the production of 76 egg embryo, right? So total, by adding these two, we get total 95 meiotic divisions. Now let us see question number 3. Here is the question: how does pollination takes place in water hyacinth? So water hyacinth is an aquatic plant. It is an aquatic plant, but it is an aquatic flowering plant. Aquatic plant and flowering, right? So flowering means the pollination is through some agents. But here let me tell you, though it is an aquatic plant, it is not pollinated through water. So this is the reason why the question is asked to you. You may be confused, or you may think that since it is an aquatic plant, so it must be pollinated through the water. But let me tell you, pollination by means of water is common among the groups of plants like the bryophytes or the pteridophytes. But in case of flowering plant, very few are pollinated through water. So their flowers are not submerged. The flowers are above the surface of the water. They are, above the surface of the water, so they are in contact with the air, right? So in this way, you can write here that in case of water hyacinth, the pollination may take place through insect or insect or wind. So the pollination in water hyacinth is either through insect or through the wind, right? So this is how pollination may occur in this case. So it is not a water pollinated flower or the plant, but it is a wind or an insect pollinated plant. Since their flower is above the surface of the water, so they are in contact with the air, so possibility that it may be either pollinated through the wind or through the insect, right? So in this way you can write your answer. Now let us see question number 4. Here the question is, name the glands that contribute to seminal plasma. So this question is from the topic male reproductive system and the accessory glands. So the male reproductive system, other than the primary organs, it also consists of some glands which includes the seminal vesicles, prostate gland and the bulbourethral gland. So the secretion of these different glands constitute the seminal plasma. So this seminal plasma contains fructose, calcium or some enzymes that help in the nourishment of the sperm, as the sperm has to remain viable and healthy until it reaches to the female reproductive system for the fertilization to happen. So in this way you can write your answer as the different glands are the seminal vesicles, prostate gland and the bulbourethral gland that constitute the seminal plasma, which help in the nourishment of the sperm for the fertilization to happen, right? Now our question is here, a snapdragon plant with violet flowers was crossed with another such plant with white flowers. Here the violet flowers was crossed with white flower. The F1 progeny obtained is pink in color, right? Now the question is, explain in brief the inheritance pattern seen in offspring of F1 generation. So in this question, since it is a 1 marks question, here you need to explain why this pattern or what kind of inheritance pattern it is. So in the question, the snapdragon flower with one is purple when it is crossed with white. Okay, so the result, this is the parental type or the parental plant with purple flower and white violet flower with violet cross with white, then the result is pink in color. When they cross, the F1 generation came in pink color. So what is the reason for this? Which inheritance pattern it is? It is the pattern of incomplete dominance. It is incomplete dominance, right? So in incomplete dominance, it means the violet color is not completely dominant over the white flower, right? So the violet color is not completely dominated over the white color. So according to Mendel's law of inheritance, here the F1 progeny should be violet in color. Okay, so the gamete will be capital V, small v. It should be violet in color. But instead of violet, it came out to be pink in color. And the reason for this pattern of inheritance is due to incomplete dominance, right? So in incomplete dominance, the violet color is not completely dominant over the white color. So you can simply write your answer is due to incomplete dominance. Okay. Let us now discuss the next question, that is to differentiate between aneuploidy and polyploidy. So these are the the chromosomal abnormality or the change in number of chromosomes. So in aneuploidy, here there is the addition of one extra chromosome or the deletion of one extra chromosome. That is our chromosome number is diploid 2n. So aneuploidy, either it can be +1, results in the number of chromosome as 47, or it can be lack of one chromosome, that is 2n minus 1 results in 45 chromosome. So this phenomena or this disorder, you can say, the change in the chromosome number is known as aneuploidy. Whereas polyploidy is the entire set of chromosome is double. That's why poly means many. Say for example, prior to cytokinesis, the number of chromosome is double. That is 46 chromosome is double. That is 46 plus 46 becomes 92. So total 92 chromosome. But due to some reason or due to the failure in cytokinesis, if the cell will not be divided into two, it will result in the double number of chromosome from 46 to 92. So this phenomena is known as polyploidy, where the entire number of chromosome is double and result in the multiple number of chromosomes in the same cell, which is called the polyploidy. So this is since 1 mark question, so you can explain it with one sentence. Aneuploidy is the change in the number of chromosome, single number of chromosome, either 2n plus 1 or 2n minus 1, or whereas polyploidy results in the doubling of the chromosome into double, or change in the number of chromosome into double from 46 to 92 due to the failure in cytokinesis. Okay, question number 7. Predict the effect of if the codon UAU coding for the amino acid at the 25th position of a polypeptide of 50 amino acid is mutated to UAA, right? Now here the question is, we have to tell the effect. What will happen if the code with UAA is replaced by UAA at 25th position? Since we know that polypeptide is a long polymer of amino acids, right? So there are amino acids which are joined together by a peptide bond. So in this way, it's a long chain of amino acid. So at the 25th position, say this is at the 25th position. 25th position, it is supposed to be the amino acid having the code UAU. Now because of mutation, this UAU is replaced. This UAU is replaced by the codon UAA. Right? Now what would happen? So we should know that, and we know that this UAA codes for the codon which are the stop codon. Right? So we know that the stop codon, there is no tRNA for the stop codon. As a result, the mRNA which contains or which consists of the code UAU will be stopped, or it will stop to translate at its 25th position. So instead of the polypeptide with 50 amino acid, the process of protein synthesis or the polymerization will be stopped at the position 25th due to this stop codon, that is UAA, right? So in this way, the protein synthesis, the process of translation will be stopped at 25th position. So in this way, you can write it that since UAA is a stop codon, so the polymerization or the synthesis of proton or the translation of protein will be stopped at 25th position, which is mutated from UAU to UAA. Now let us discuss the next question, that is differentiate between proinsulin and a mature insulin, right? Proinsulin and mature insulin. So in human, the insulin is synthesized in its an immature form, or like the proenzyme, or an immature form which is known as pre-insulin, which has an extra stretch of C-peptide, right? So which needs to be processed. So the proinsulin has got an extra stretch. You can write your answer as an extra stretch of C-peptide which is absent in a mature insulin. Which is absent in mature insulin. So you can write here, the proinsulin is the unprocessed, or it's an immature insulin which is synthesized in its immature form, or you can say in its inactive form, which needs to be processed, and that contains an extra stretch of C-peptide which is absent in mature insulin. Means the pro-insulin gets converted into a mature insulin which does not contain the C peptide. So that is the difference between pro-insulin and a mature insulin in human.